Okay, here we go with homologous recombination. Here I am. So, we use homologous recombination when we need to repair double-stranded breaks in DNA. This slide lists a few of the causes of double-stranded breaks. You can see anything that damages DNA has the chance to cause double-stranded breaks. That could be oxygen radicals, reactive oxygen species, ionizing radiation, like the extreme radiation you get if you're doing, when people get radiation for cancer, or stalled replication forks, and that could be the things that stall replication forks are all other types of DNA damage that perhaps weren't carefully repaired. And of course, sometimes we have enzymes that we can use in lab or even in vivo occasionally that we introduce double-stranded breaks on purpose. Usually in lab, we do this a lot in lab, and we'll talk about that later. Okay, so here we have a DNA, two strands, two double helices next to each other. And we have our double-stranded break here starting at the top, okay? There's our double-stranded break. And so the basic steps that initiate homologous recombination are shown on this slide. So the first thing that has to happen is we need some nuclease activity going on here. Doesn't show very well. That one. Chewing back from each end. And this will be a couple hundred base pairs or so. It can be a pretty long region that they chew back. The result of that, of course, is some single-stranded DNA here and here. Single-stranded right here. We got a single-stranded there. And we'll talk about in a minute that we actually would expect that'll be protected by some proteins in a second. The next step in terms of the DNA, though, is what's called strand invasion. So one strand of DNA that is activated and will come and disrupt the second strand. That's this, and this strand in blue is the one that's going to be used for repair. It's the repair template. And so this red strand, the single-stranded protein-covered single strand, has come in here and disrupted this intact blue strand, and this structure is called a D loop for disruption. Disrupted loop. And then once that's in there, DNA polymerase, I'll see if I can draw it the way the textbook draws it. Not probably very good. We'll use that single-stranded end as a primer and start filling in some of the missing nucleotides. And this can happen on both strands eventually until we get to a holiday junction. One of them is shown right here. So once we've created this holiday junction, or it's really a double holiday junction, so we have that kind of this repair bubble going on. And so that's now in the middle of the slide here. Here's one of the holiday junctions, and then you can see a second one is formed up here, and eventually this could get ligated together. This thing needs to be resolved. And there's a couple different ways that can happen. So in fact, there's 3 different ways that can happen. They're summarized on this slide. Of course, we're going to go through them one at a time. So option 1 is at the top. We're going to go through that first, which is called synthesis-dependent strand annealing. And then we're going to go through options 2 and 3 after that. Okay, so let's talk a little bit more about what happens during strand invasion, though. So what we have here, we have our double-stranded break, and we see the region that's been chewed back. So there, double-stranded break, check. And the first thing that happened is RPA came in to protect that single-stranded DNA, and I hope you guys remember what RPA is. It's a single-stranded binding protein. That's the eukaryotic one. It protects all single-stranded DNA. The E. coli version is called SSB. Okay, so we're protecting our single-stranded DNA that got exposed by first the double-stranded break and then degradation of some of the extra DNA there. The next player to come in to help us out here is called is RAD52 here. And what RAD52 does is zips along here and dissociates RPA. It associates the single-stranded binding protein. But as it's doing that, another guy is coming into play, and that's RAD51. And RAD51 is filling in where the single-stranded binding protein is, because RAD51 has a special job in initiating strand invasion. So whereas RPA can just protect the DNA from random degradation in the cell, RAD51 is part of the DNA repair machinery that's associated with the homologous recombination. And what that does is allow the next steps to occur. And I just want to point out that right here I have a link to a video which has some nice animation kind of showing what happens with the strands and maybe help you visualize it a little bit better. It focuses more on the strands and less on the proteins. Right now I'm talking about the proteins, so it's a nice video to look at from Cambridge, I think. Okay. So this is what happens after after RAD51 has bound to single-stranded DNA. In this case, the example we're looking at is REC-A. REC-A is from E. coli. It's an ortholog. It does the exact same thing. And you can see it's literally coated the strand of it forms a filament. Okay, and that protects it protects the linear single-stranded DNA, keeps it linear, and protects it when it forms a long filament on there. Many copies of it are bound along there. This slide just shows what REC-A and RAD51 look alike. So REC-A is on the left of the slide here. This is the E. coli ortholog. And you can see maybe right in here where the DNA is kind of bound. It's kind of maybe that's got a little hand-like structure too. I don't know. We're not going to get too much into the structure. I just wanted to show you that. And it's relatively similar to RAD51. They are considered orthologs, and RAD51 is from humans. And there's all variants of RAD51 throughout other eukaryotes as well. And again, you can see a little piece of DNA bound in there. Okay. So they form a filament, and that allows for this thing called strand invasion to occur. So just to kind of review quickly, we had our double-stranded break. We had some nucleus activity chewing back, and then we had the first thing, we had the single-stranded binding proteins bind. And the nice thing about this slide, it shows both the names of the E. coli proteins and some eukaryotic ones. So there's single-stranded binding protein, there's RPA. So we're protecting the single-stranded DNA. In comes RAD52, which displaces RPA. The equivalent of that in E. coli is REC-O. Does the same thing. So it displaces the single-stranded binding protein and allows for the strand exchange protein to bind. Strand exchange protein, the one that forms the filament, is RAD51 in humans, REC-A in E. coli. And now, once that is coating the DNA, as you can see it here, it now is capable of doing strand invasion. So it's doing a little more than just single-stranded binding protein. It allows this strand invasion event to occur. It gets in there. You can see the D loop is formed, which is this entire region here is a D loop, displacement loop. And then replication will extend off the linear end of we'll use the single-stranded DNA as a primer and start filling in some missing DNA. So now we want to resolve this thing. So here we are. We're now back to the top of the slide I showed you earlier, just ignoring the bottom. So it's option 1. What happens? This is called synthesis-dependent strand annealing. You can see that right here, the name. And basically what happens is DNA polymerase just keeps going. and going, and makes copies from the blue intact DNA helix. Double helix is acting as a template, and we've now had strand invasions, and we're extending off each of the loose 3' ends of our single strands and extending and completing our double-stranded, filling in the missing DNA sequence. Then what's happened at the end now, we have this double crossover event here. These are called Halliday junctions, and there's 2 of them, and this thing needs to be resolved. And one way that can be resolved, it can simply be dissolved, actually, I should say. It can be resolved or dissolved, and one of the solutions is called dissolution. And that is what happens when we use the enzymes topoisomerase and Bloom's helicase. So this is dissolution. And basically they work in conjunction with each other. Bloom's helicase, you can picture it kind of twisting around. And as it's, let's say that's kind of coming out of the plane of the screen, and it's kind of twisting around to unwind this, but at the same time topoisomerase is on the other end there nicking and relieving torsional strain that's accumulating as the Bloom's helicase works its way through the structure. And I guess they actually be proceeding in this way because we're shrinking this bubble until we completely resolve it. And when it's finally resolved, we've got 2 complete separated strands of DNA. The 2 copies of the blue strands are back together. There was no exchange of any genetic information, and then we've repaired the gap that we had here. Okay, so now we've talked about how we can dissolve Halliday structures. And now we're going to talk about how we can resolve them. And resolve them requires some enzymatic action within this area here. One of the enzymes, the first one to be discovered, was RUV1, and it is known as a resolvase. So it has to be able to cut DNA and twist it and rejoin it in context of the very specific structure that's formed at Halliday junctions. Resolvase. There we go. Okay, so this slide actually shows all 3 possible outcomes of homologous recombination. We already talked about dissolution, which is in the first panel here. I do want to point out that this is only in eukaryotes. And there is actually a disease called Bloom syndrome that's associated with this BLM gene. Okay. Now we're going to talk about the resolution, though. So, not shown on the slide is the fact that RUV-C is the E. coli equivalent of Gen 1. So you can, whatever we see in this Gen 1 panel is also happening when there's RUV-C present. And then this is considered, so this is called, this middle panel is considered the canonical resolution pathway. I'm just writing it for short. This is the non-canonical. Discovered a little bit later. Not quite as accurate, or not quite as robust, I should say, but it can help in a pinch. So the most important protein you need to know about on this panel is Gen 1, which is, again, does the same job in humans that RUV-C does in E. coli. So we talked about how RUV-C is the resolvase of the Halliday structure, so is Gen 1. And then what we need to talk about is the fact that depending on where the cuts are made, we're either going to have exchange of genetic information or not. And this is the best way to think of it is that if the same strands are nicked in all the locations. So let's see which that would be. There actually be 2 top arrows like this. So if we had another pair of top arrows like that, I'll make them in a different color. It would be non-recombinogenic. That's because we'd go from here to here, and then from here to here. But when you nick alternate strands, that's when you have recombination. And there's 4 locations that can be nicked in any holiday structure, but we only need to nick 2 of them at any particular time. And so you either nick north to south or east to west. And so if you nick north south and north south, it'll be non-recombinant. And if you nick north south at one junction and east west at the other junction, it'll be recombinant. And then with this non-canonical mechanism of resolution of the holiday structures, you can see there's slightly different locations where the nicking's occurring. It's the same kind of phenomenon. Here we have essentially northeast to southwest, or whatever you want to call it. They're either the outside strands and the inside strands, or you can have all the outside strands or all the inside strands nicked. So depending on which combinations of strands are nicked, you're going to have either recombination or non-recombination as you resolve the holiday structure. This slide just shows you the 2 enzymes, the 2 resolvases I was just talking about, the 2 canonical resolvases I was talking about. Canonical because ROB-C was recognized first, and then for many years people were looking for the eukaryotic resolvase. Canonical. Okay. And so this just shows their structures. And it turns out they are actually, even though they have the same function, they aren't actually orthologs. And that's because in our cells, we have a couple more places where we need to facilitate DNA repair. E. coli kind of has it simple. We have meiosis, we have mitosis, we have a full cell cycle and kind of regulation within that. This slide summarizes the proteins involved in bacterial recombination repair, and I've just highlighted the ones I want you to know, which are Rec-A, Rec-O, ROB-C, and hopefully you remember these guys from DNA replication. So Rec-A is required, it's the filament, it forms a filament and is required for strand invasion. Rec-O facilitates the binding of Rec-A. And ROB-C is the resolvase. Okay, well, this is just another summary of what we were talking about. So we have our double-stranded break, we have resection, we have strand invasion that's mediated by Rec-A or Rad51. Okay, we have DNA synthesis, we have double holiday junction there, and then we'll go to our different options. So again, we have the dissolution option, which is shown here, or the in this case, they're calling it symmetrical or asymmetrical. We call this canonical. In our discussion, we're going to refer to this as the canonical. Resolution. And now you can see on this side both ROB-C and Gen 1, whereas asymmetric or non-canonical is not a pathway that's present in E. coli. It's just this is eukaryotic only. And also dissolution is eukaryotic only. Okay. This slide summarizes the proteins I want you to know. In particular, so we've talked about some of them we already knew about. Our DNA protection, our single-stranded binding protein is here. This strand mediator, or the one that kind of, so Rad52, Rec-O interact with the single-stranded binding protein, disrupt them, and allow for the strand invasion proteins to bind, right? And that's how we've created our D loop. And then if we have dissolution happening, that's mediated by topo3. And ROV-A and ROV-B, or these SLX proteins, are involved in stabilizing the holiday junction. And then these are our resolvases down here. Canonical resolvases. Okay, so that is homologous recombination. Next up, we're going to be talking about non-homologous end joining. And I'll preface that by saying right now, this is actually pretty accurate and robust. And it's template based, so it's not guesswork. In the case of non-homologous end joining, we're going to see it's actually not too accurate, but sometimes it's necessary. And that's where we are.